Hello and welcome. This video will cover acids and bases concentration by titration method. In this experiment we are going to do 4 trials, each trial with 2 different chemicals, 1 acid and 1 base. Always we are going to add acid to this main beaker, and we are going to use the root available here to add small portions of the base. The volume we are adding depends on the pH readings, so always refer to your lab sheet procedure in order to know how much exactly you have to add at each step. Before we start, we'll need to make sure that the pH meter probes are ready for measurement by washing them both, and you can always start this experiment with pH meter calibration. We'll need to get both probes inside this small beaker. We are going to add the acid first, and our first trial is going to be HCl. While doing this experiment, you will need to make sure that you are covering the minimum safety requirements, which will be safety goggles, gloves, and lab coats. Always be careful while dealing with chemicals. Then we are going to use a funnel to fill in the broth likewise. Then fix it on the available clamp. So I'm going to do that right now. Now we can start by adding small portions based on your lab sheet. We can get 10, to avoid any chemical spillage on the workstation. You can always look into the grades available and know exactly how much you are adding. You will need to note down your starting point, and you will need to note down your starting pH. You are going to start with adding 1 cubic centimeter at the beginning, watching the pH changes. It's going to change slowly, as you can see in there. Based on the changes, by changes we mean that the difference between initial reading and the next reading, you are going to decide how much to add each time. You will need to keep doing so, taking in consideration the volume added and the pH changes. Always refer to the volume measurement while adding, and you can see the liquid level is changing slowly in here based on how much we are adding. You can always control this valve to slow or make it faster. You need always to make sure that the pH probe is submerged with a chemical. If it was floating or outside, that will give you wrong readings in there. So make sure the probe is submerged. Keep adding, follow your procedure on the quantities required, and that will finish one trial. Next, you will need to clean everything and start with the next chemicals available. Remember, always before changing with chemicals, you will need to run the pH probes for temperature and pH. So this is basically it. Thank you for watching.